Good morning all of you. Today the topic of the class is development of nervous system. The nervous system is derived from ectoderm except blood vessels and some neuroglyals like microglia which are mesodermal in origin. And a specific type of ectoderm which gives rise to nervous system is called as neuroectoderm or the neural ectoderm. This neuroectoderm will differentiate into three structures. First is the neural tube which give rise to central nervous system. Second one is the neural crest cells which forms the peripheral nervous system. And third one is the ectodermal placoids. They form the cranial sensory ganglia, hypophysis and the inner ear. So ectoderm divides into neural tube, neural crest cells and the ectodermal placoid. Neural tube forms the central nervous system, neural crest cells forms the most of the peripheral nervous system, and ectodermal placoids are forming the cranial sensory ganglia, hypophysis cerebri and the inner ear. Now we come to the neurulation. Neurulation is the process of formation of neural tube, and it starts about 16th day of embryonic life. The ectoderm which is lying over the notochord, this one yellow one is the notochord, and the ectoderm which overlies over it is called as neural plate. This central part of the neural plate is depressed, and this margins of the neural plates are elevated, that are called as neural fold, and central depressed part is called as neural groove. So neural fold, these are the neural folds, they gradually move towards the midline and they finally fuse to form the neural plate. And here you can see this is the diagram showing development of the neural tube. In this one is the notochord, and ectoderm over it is called as the neural plate, and the margins of this neural plate are called as neural fold, and the central part of the neural plate is depressed to form the neural groove. Now the neural folds are moving towards each other and they will finally fuse to form the neural tube. So thus the formation of neural tube occurs. And on the margins of the neural fold, the purplish structure that is called as neural crest cells. They will finally form the neural crest cells, and they are separated from the ectoderm. So these neural crest cells has been separated from the surface ectoderm. This is the ectoderm, and they has been separated from the ectoderm. So these neural crest cells are present between neural tube and the ectoderm, and they lie dorsolateral to the neural tube. So neurulation is the process of formation of neural tube, and which starts around the 16th day of embryonic life, and when the formation these neural fold fuses, the fusion starts in the middle part, and then it proceeds further in the cephalic and caudal direction. But so the fusion at the cranial end and the caudal end of the neural tube is somewhat delayed, and that lead to formation of some small openings that are called as neural pores. So at the cranial end, formation of cranial neuropore or the anterior neuropore, and at the posterior and caudal end, formation of caudal neuropore or the posterior neuropore. So these are the sites where the neural folds has not been fused at cranial and caudal end, and these openings are called as neuropores. By these anterior and posterior neuropores, this neural tube is connected by a cavity, that is amniotic cavity. This white structure is the cavity is the amniotic cavity. This arrow is showing the amniotic cavity, and through these cranial neuropore and the caudal neuropore, this amniotic cavity is connected with the neural tube. And what is the function or what is the reason of this connection? Because this neural tube get nourishment through this amniotic fluid. Now this anterior neuropore closes at the middle of the fourth week and posterior neuropore will close at the end of fourth week. And we will say anterior neuropore closes at 18th to 20th somatic stage, and posterior neuropore closes at 25 somatic stage. So the closed cranial end of the neural tube will persist as a lamina terminalis. So cranial opening is called as anterior neuropore and caudal opening is called as posterior neuropore, and this anterior neuropore will close at the mid of the fourth week or around 25th day at 18 to 25 at 18 to 20 somatic stage, and posterior neuropore will close at the end of fourth week at about 25 somatic stage, and these neuropore will connect the neural tube with amniotic cavity. Now formation of this process was neurulation. Now we come to the neural crest cells, that is very important type of cells and also important for exam point of view also. So these neural crest cells, as we have discussed earlier, that when the neural folds come closer to each other, cells which are present at the tip of neural fold, these are the neural fold, and the cells which are present at the tip of neural fold are called as neural crest cells. These neural crest cells, they are initially they lie in between the neural tube and the surface ectoderm. This yellow one is the neural tube and this is surface ectoderm. So they lie in between these two structures, and they lie dorsolaterally to the neural tube. These neural crest cells will give rise to dorsal root ganglia, sensory ganglia of the cranial nerves, autonomic ganglia, and the adrenal medulla, chromaffin tissue, melanocyte, and the Schwann cells. So these all structures, they are developing from the neural crest cells, and these neural crest cells are present at the tip of the neural fold. Here the purplish structure is showing the neural crest cells, and they will get separated from the surface ectoderm and they will lie in between the neural tube and surface ectoderm and give rise to multiple structures, important structures. These are dorsal root ganglia, sensory ganglia of the cranial nerves, autonomic ganglia, adrenal medulla, chromaffin tissue, melanocyte, and Schwann cells. Now we come to the neural tube defects. These are the neural tube defects, are the defects of the spinal cord which are resulting from the abnormal closure of the neural folds. And we know that the closure of the neural folds occur during around 4th week of development, 3rd to 4th week of development. Closure in the middle will start in the 3rd week, and anterior and posterior neuropore will close in the 4th week. And if there is abnormal closure of these neural fold, that will lead to development of neural tube defects. And if during many studies have shown that if the pregnant female is taking folic acid supplements before conceiving and during first 3 months of the pregnancy, that will reduce the incidence of neural tube defect. And some drugs some anticonvulsant drugs like valproic acid and other drugs also, they may lead to abnormal development of neural tube, or we can say that will lead to neural tube defect if they are taken during 4th week, during 1st to 4th week of the pregnancy. And these neural tube defects, they involve meninges, vertebrae, muscles, and the skin. One neural tube defect is spina bifida is general term which is used for the neural tube defect which is involving the spinal region. And this spina bifida consists of splitting of the vertebral arches And there are two types of spina bifida. First is the spina bifida occulta and the second one is spina bifida cystica. Here you can see the spina bifida occulta. It is a mild defect or the minor defect in which there will be defect in the vertebral arches. So, and this area of the skin is covering that overlying area, and it doesn't usually involve any neural tissue. So, spina bifida occulta is defect in the fusion of the vertebral arches, and this area is covered by skin, and generally there is no involvement of any neural tissue, and it most commonly occur at lumbosacral region, and this area is usually marked by a patch of hair. Here you can see the patch of hair overlying this affected region. And why this defect occurred? Due to lack of fusion of the vertebral arches. So, spina bifida occulta is the minor form of the neural tube defect that involves the lumbosacral region L4S2S1, and that is due to failure of the fusion of the vertebral arches, and this area is covered by skin and the tuft of hair is present here. Now coming to the spina bifida cystica. It is a severe neural tube defect in which neural tissue or the meninges, they will protrude through the defect in the vertebral arches, and skin will form a cyst like sac, and most commonly it is also present in the lumbosacral region, and it will also result in neurological deficit. This sometimes this cyst is filled with fluid, it is filled with cerebrospinal fluid or fluid filled meninges, they will protrude through the defect, then it will called as spina bifida with meningocele. Means cyst is filled with fluid and meninges, and they will protrude through the defect in the vertebral arch. And in some cases neural tissue will also come into this cyst or in the sac, and then we will call it spina bifida with meningomyelocele. Myelio means spinal cord and meninges. So, spina bifida with the meninges and spinal cord, then it is called as meningomyelocele. Here in this diagram, first diagram is showing the spina bifida occulta. In this the area is covered with skin and the tuft of hair are present here, and no defect on the surface. Second one is the meningocele. In this there is a cyst like swelling and only the meninges are bulging through it. And third one is the meningomyelocele. In this condition, this spinal cord and meninges both are protruding through the cyst. And sometimes this neural fold will not elevated, but it remain as a flat mass of the neural tissue. So that will called as spina bifida with rachis or myeloschisis. When the neural fold is not elevated but remain as flat mass of neural tissue. And the fourth diagram you can see rachis, fourth and fifth condition. In this the neural this area is not elevated but neural tissue is lying in the gap. Here the fifth diagram also showing the folded neural tissue in the gap. So these are the neural tube defects. Now anencephaly or the cranio-rachis. The failure of fusion, we have discussed that neural pores, anterior neuropore will close at around mid of the fourth week. If there is fusion, failure of fusion of cephalic part of neural tube, then that will lead to cranioschisis or the anencephaly. In this condition, there will be defective development of vault of the skull, and so that will lead to brain is not covered, and so brain is represented by a mass of degenerated tissue and it will be exposed on the surface. Spinal cord is open in the cranial region. This is a severe type of neural tube defect, and it includes acrania, means absence of cranial vault, so that will lead to cerebral regression, and there is enlargement of the foramen magnum, and you can see a surface depression in the sacral region, that is sacral dimple, that is marked by a arrow in this diagram. So appearance of a child is with prominent eyes bulging forward, and chin is continuous with the chest due to absence of neck. So this is anencephaly or rachis in which the cephalic part of the neural tube fail to close, and leading to defective development of the vault of the skull, and brain is exposed to the surface, so it is degenerated mass, and spinal cord is also open in the cervical region, and you can see a sacral dimple. Now rachis, that is also a severe form of spina bifida, and in this condition there will be incomplete closure of the caudal neuropore, and this is also associated with the due to defective development of the vertebral arches. So if the neural tube fail to fuse in the cranial part, then it is called as anencephaly, and if there is incomplete closure of the neural tube in the caudal part, then it is called as rachis. And what is in rachis? Characteristic feature is failure of dorsal portion of the vertebral arches to fuse with each other, and usually it is also localized to the lumbosacral region, and the neural tissue is widely exposed on the surface. Here you can see the neural tissue exposed on the surface.